Okay, so we are going to discuss chapter 7 part B. We are going to start with question 1. So our last minggu lepas your punya group tak ada kan? Kita cuti, right?
Yes.
So kita pergi atas ke bawah ke atas? Nak pergi dengan Fikri dulu ke? Sebab saya tak ingat tu awak punya group rotation. Fikri dulu ke Yasmin dulu?
Tak kisah mana-mana.
Tak kisah.
Kita pergi dengan Fikri dululah. Alright? Okay. So can you do question number 1 please?
Yes. So question number 1. Figure 3 shows cell that lining the respiratory tract. Staphylococcus aureus bacteria are seen sticking to the mucus secrete. So name structure M. Structure M is cilia.
Okay, Cilia. So thank you Fikri. Ainin, number 2.
State function of the mucus in figure 3. My answer is mucus tract pathogens.
Trap pathogen. So in this case is the bacteria lah kan. So you can either cakap trap pathogen or to be specific as in the question to trap the bacteria. Okay. And then also the mucus will also contain lysozyme. Okay, lysozyme that will destroy the bacterial cell wall. So the mucus is produced by what cell lining the respiratory tract? Sel apa yang akan secrete mucus ni? Apa nama sel ni? Ainin. What is the name of the cells secreting the mucus that lines the respiratory tract?
Goblet cell.
Okay, goblet cell. Fikri, balik pada cilia tadi. Kan, what is the function of the cilia actually?
Cilia contract.
Apa dia? Mucus, dia dah trap bakteria kan? So this, is it remain in your respiratory tract? Tak boleh kan? So that is the function of the cilia to swipe the mucus containing the contaminant upward so that it enters into the glottis and then masuk ke awak punya digestive tract, which is it enters into your esophagus. Awak punya respiratory tract, trachea, and belakang dia ada esophagus. Antara trachea dengan esophagus tu akan ada pertemuan kan, awak punya rongga kat belakang ni akan ada pertemuan junction where the cilia containing the mucus containing the bacteria, that combination will be swiped upward by the cilia until it is swallowed into your esophagus and enters into your stomach to be destroyed by your stomach acid. Okay, so dia tak boleh nak remain dalam awak punya trachea tu je kan. So dia kena swipe by the mucus upward into the glottis and then swallowed, enters into the esophagus, swallowed into your stomach. Okay. So that is the function of the cilia. Bukan cili cilia. Lupa nak tulis A. Okay. So thank you to both of you, Fikri dengan Ainin. So Isha, the next question B please.
B. Fever of elevation of body temperature is a common response to infection. Give one benefit of fever. Inhibit the growth of some pathogens.
It inhibit the growth of pathogens, satu markah. So it also has several other benefits. Okay, satu promotes phagocytosis. Okay, it can also promote phagocytosis. accelerate tissue repair, and also stimulate infected cell, okay, which is infected by the virus, to produce interferon. Okay, so the interferon produced will promote the process of phagocytosis lah by the macrophage, for example. Okay, so betul tak? Dia ada dalam nota awak ni, dia ada bagi dalam table. Fever dalam mana? Dalam second line defense Fever dalam second line defense, innate immunity, dia termasuk bawah inflammatory response. So the benefit of fever yang tadilah promote phagocytosis by phagocytic cells and then inhibit growth of some pathogen by stimulating the liver and spleen to store iron so that blood iron level is reduced. Kenapa bila awak demam, liver ni has to make sure that the iron level is reduced by storing the iron. Kenapa nak kena make sure the iron level is reduced? Saya nak tanya. Pathogen, they are living organism, right? Okay, for any living organism, apa yang menyebabkan organism tu alive? Disebabkan oleh aktiviti metabolism yang berlaku dalam sel tu kan. Apa yang membolehkan aktiviti metabolism berlaku dalam sel tersebut? Adanya enzim, kan, enzim. So, kita dah belajar dulu masa chapter semester 1 enzim. Enzyme, in order for an enzyme to work, they have to have a cofactor. Okay, one of the example of cofactor adalah iron, kan? Ion. Metal ion. Okay, metal ion. So iron is they function as cofactor for enzymatic activity to occur in pathogen. So by reducing the iron level, so this reduces the metabolic activity inside this pathogen. Faham tak? Boleh So that's why dia kena reduce the iron level. Boleh?
Boleh, madam.
Okay. So ingat inflammatory response. Kalau demam, dia adalah systemic inflammatory response. So maksudnya seluruh badan awak panas. Okay. So that is the whole of your body increases its temperature. That is fever. Okay. So the next question, C. Anas? Anas ada?
Oh, yes sir. Ada.
Question C, please.
Question C. Cell X. Cell X ni apa? Cytotoxic cell. Okay.
Apa ni? Kalau C yang cell X is what?
T cytotoxic cell.
X. Kalau Y?
T helper cell.
Kita tengok soalan ni Figure 4 shows antigen recognition by immune cell. Awak tengok kalau sel yang besar ni dia adalah macrophage. Okay, so macrophage, when the macrophage internalize the virus, it engulfs the virus, dia akan jadi sel yang kita panggil sebagai apa? APC, antigen presenting cell. So kenapa kita panggil sebagai APC? Because this APC cell, okay, the macrophage that becomes the APC present the antigen molecule, which is the virus that is degraded inside the macrophage, degrade jadi antigen fragment. Dia akan present guna MHC class 2. MHC class 2. Okay, MHC class 2. So dia akan present kepada sel apa? Sel X. Sel X ni tadi awak cakap cytotoxic T cell. Fungsi cytotoxic T cell adalah untuk destroy infected body cell. Bukan dia bukan untuk destroy APC. So supposedly this one is what?
T helper.
Okay. T helper cell. Okay, T helper cell. So cell X is T helper cell. Kalau sel Y dia sebenarnya adalah yang cytotoxic T cell ataupun T cytotoxic cell. Okay, so this is infected host cell, any cell that has nucleus. So when virus enters into the host cell that has the nucleus, so the virus will be fragmentized into antigen fragments, and then dia akan bind dengan MHC class 1. Kan, so dia akan present guna antigen MHC class 1, dan the antigen will be presented using class 1. Dia akan present kepada cytotoxic T cell, and once you have this interaction, so the cytotoxic T cell will destroy this infected body cell, okay, exposing the virus to the circulating antibody, for example. Okay, so yang ni awak kena tahulah. So ini sel X adalah helper T cell, sel Y adalah cytotoxic T cell. Cuma dalam exam tolong tulis nama penuh dia, jangan short form macam ni so it is safer for you to write the full name of the cell. Okay,
Okay, madam.
so
Thank you.
okay, so Anas, thank you. So next, Isha.
Saya dah, madam.
Dah. Oh, tadi sebab Fikri dia keluar. Oh, tak pula. Oh, dah Aisyah. Okay, next Hanan.
Based on figure 4, describe the activation of cell Y. So, cell X, the T helper cell, will bind to the antigen MHC complex on the surface of the APC. Then the binding of the T helper cell will trigger APC to secrete cytokines which stimulate the T helper cell to produce its own cytokines and produces an activated T helper cells. The activated T helper cell then secretes cytokines to activate cell Y T cytotoxic cell.
Okay, betul lah. You have the whole story there, kan? So you got the full 3 marks lah. So basically, kan? Describe the activation of cell Y. So cell Y adalah cytotoxic T cell. It has to be activated. How to activate this cytotoxic T cell? Dengan cara yang ni dulu. Or explain yang macrophage ni dulu. Okay. Point yang pertama, macrophage engulf the virus, and then the macrophage will degrade the virus into antigen fragment, present using MHC class 2, present kepada siapa? Helper T cell. Once you have this interaction, the macrophage akan secrete signaling molecule which is cytokines. So the cytokines akan activate helper T cell. Helper T cell that is activated secretes dia punya own cytokines, activate more of the helper T cell, undergo clonal selection, jadi apa? Memory dengan effector. This helper T cell punya cytokines akan activatekan cytotoxic T cell. The cytotoxic T cell, once it is activated, it also undergoes clonal selection to produce effector, dan juga memory. Okay, memory cytotoxic T cell. Yang awak kena take note adalah yang secretion of cytokines ni. Kedua-dua APC dan juga helper T cell will produce this cytokines molecule. Okay, so So awak start dengan cerita macrophage sampailah cytokines ni boleh diaktifkan semula, diaktifkan. Boleh Basically bila awak tengok gambar rajah ni, okay, gambar rajah ni, you should be able to tell the story what happened in this diagram. So basically yang ini sebab dia melibatkan cytotoxic T cell, it involves the process of activation of cell mediated immune response. Sebab dia melibatkan cytotoxic T cell destroying infected body cell. Okay. So 3 markah. So awak kena cerita the full story which is explained by Hanan tadi tu. Okay. So next, Ishraf D.
Madam tu kelas 9.
Oh, kenapa ada Ishraf sini? Izzat Daniel.
Explain the changes of Ahmad's antibodies after the second vaccination. The memory B and T cell recognize the antigen present. Second point, B and T cells will rapidly activate immune response to produce a huge amount of antibody to fight the antigen.
Okay. Basically, when you look at this diagram, apa yang awak boleh cakap kat sini? What is the difference between these two peaks?
High concentration of antibodies after the second vaccination.
Okay. Dalam acquired immunity, tadi bila kita tengok kan, bila helper T cell activated, bila cytotoxic T cell activated, kan, dia menghasilkan effector dengan memory. For the first time infection by the virus, in this case bukan infection by virus tapi vaccination. What is vaccination? Vaccine lah kan, you insert vaccine into your body. What is vaccine? Vaccine tu it can be the virus yang dah dilemahkan, contohnya virus yang dah dilemahkan, bila dia masuk dalam badan awak, that virus will not trigger an immune response because it is dead, for example, or dia dah dilemahkan lah. Ataupun macam contohnya yang terbaru apa, you insert mRNA kan. So by inserting this molecule that does not trigger your immune system, that will not get you sick. Dia bukan, purpose vaksin ni bukan nak menyebabkan awak sick, tapi dia nak masukkan this foreign molecule against that specific virus to trigger your immune system. For the first time, this foreign molecule enters into your body, dia akan menyebabkan primary immune response. Okay, primary immune response. Primary immune response, it takes longer time sebab proses tu dia nak memastikan proses tadi macrophage datang, engulf, and then activate helper T cell. Helper T cell pula akan secrete cytokines, membahagi lagi jadi effector, jadi memory. Lepas tu dia akan secrete cytokine untuk activatekan the effector and memory cell for T cell and also for B cell. So cerita dia panjang. So that's why it takes longer time. Okay, it takes a longer time. Tapi bila dah terhasilnya memory cell tu, when you are infected again with the same virus, or in this case for the second vaccination, your body has already produced the memory cell. Okay, so when you insert the vaccine into your body, kan, this foreign molecule in your body that does not get you sick, but it is just to trigger your immune system, your body has the memory cell. So once your body has the memory cell, your body can straight away, for example, produce antibody against that specific virus. So that's why kita ada peak terus kat sini. This is what we call it as the secondary immune response. Okay. Okay, secondary immune response. Okay. So tadi awak cakap tadi apa, antibody concentration was higher ke apa tadi? Tadi siapa? Izzat
Memory B and T cells will trigger the secondary immune response and produce a huge amount of antibody.
Betul lah, boleh lah dapat bolehlah dapat 2 markah. Okay, bolehlah dapat 2 markah. So asal kalau awak cakap pasal memory cell producing antibody in high concentration, you have the 2 marks. And also you can also include in your answer tadi kan, once you have the memory cell will rapidly proliferate. It will divide many times. Okay, many times proliferate into new B cell and also plasma cell. The plasma cell that actually produces the high concentration of antibody. So the production of antibody will take less time and produce rapidly. So that is another point that you can also include in your answer. Production of antibody will take less time and produce rapidly due to the presence of the memory cell. So therefore, after second vaccination, memory B cell already recognize the antigen, or it is exposed, okay, to the same antigen. Okay. So itu awak kena tahulah kan. So kalau dalam nota awak dia bagi contoh straight infection by pathogen, tapi ini soalan applied. Instead of pathogen, dia bagi awak vaccination. So benda yang sama. Okay, benda yang sama je. Sebab vaksin adalah virus yang dah dilemahkan ataupun component of the pathogen that does not get you sick when it is inserted into your body lah. Okay. Okay, so next, E. Hazim, please.
Question E. Skin graft is used to treat burn patient where skin is removed from one area and transplanted to another area of the same patient. Explain why the transplant skin is not rejected. Because the skin will act as barrier that prevent growth of pathogen.
Tak. Macam mana skin boleh prevent growth of pathogen? Kita tengok soalan ya. Skin graft is used to treat burn patients where skin is removed from one area and transplanted to another area of the same person. So kalau macam contohnya awak terbakar kat kulit tangan awak yang rosak. So instead of getting a skin from another person, you take your own skin, contohnya dari peha, and then you put onto your arm. Kan? So that is skin graft lah. So the skin is still from the same person. So kenapa kaedah tu lagi efektif in to make sure that tak ada rejection by your body. Cakap kat sini kan, explain why the transplanted skin is not rejected. Banding awak ambil skin orang lain, awak letak kat tempat yang luka, yang rosak, terbakar tadi tu. Kenapa using your own skin will reduce the chance of rejection? because the skin is from the same person. So when the skin is it comes from the same person, kita belajar, awak dah belajar kan? Kalau all nucleated cell, dia punya marker dia adalah MHC molecule, kan? MHC ni dia bertindak sebagai self marker. So maksudnya sebab dia ambil pada skin yang sama, so MHC molecule dia adalah sama, same MHC marker. Boleh tak kat sini? Mana tadi? Because the skin has the same, kan, same MHC marker. Okay, MHC marker 1 mark. Boleh dapat kat situ. Ataupun the transplanted skin has the same self antigen. Okay, has self antigen or same antigen as the damaged skin. Dia daripada orang yang sama je kan. Kulit daripada peha transfer ke tangan lah contohnya. Okay, has self antigen ataupun same antigen as the damaged skin. It comes from the same person pun boleh juga. It comes, the skin, the transplanted skin comes from the same person. Kalau ambil pada skin orang lain, obviously MHC marker dia will be different, kan? So dia dah considered sebagai non-self dah. So your body has the kat situlah peranan punya immune system. Dia akan detect benda tu sebagai foreign, so dia akan attack contohnya. So dia akan reduce the successfulness of that procedure tu. Boleh faham tak?
Faham?
Okay.
Faham?
Zania, kejap ya. Okay, so kita sambung dengan soalan nombor 2. Question number 2. Naiman, A.
Describe how mucous membrane protect human respiratory tracts. The answer is pathogen present in inhaled air are trapped in the mucus. Then the cilia sweep the mucus upward toward the glottis to swallow into digestive tract.
Okay, dapat 2 markah. So you have the 2 full marks. Maybe before you mention mucus tract pathogen, you can mention mucus produced by the goblet cell pun boleh dapat markah. Okay, mucus produced by goblet cell or the lining of the respiratory tract or the epithelial cell will have cilia. Okay, cilia on its surface. Tu pun boleh dapat markah. Okay, so ada 3 point lah kat situ. Right. So thank you, Naiman. Next question. B. B1.
B1. Protein U is interferon molecule.
Interferon molecule. Interferon molecule, sekejap. Okay. Interferon molecule ni, if you look at this diagram, innate immunity of which line? Innate immunity kita ada 2 line. First line and second line. For this diagram, for this mechanism, it falls under which category?
Second line.
Second line defense. So kalau tengok kat sini, innate Innate immunity dia ada dua lah. First line and second line. First line adalah external defense, skin, mucous membrane, mucous secretion tu semua first line. Tapi kalau yang dah masuk dalam badan awak, bila dia melibatkan phagocytes ataupun inflammatory response ataupun natural killer cell ataupun in this case the production of interferon, so itu adalah second line defense ataupun internal defense. Kalau interferon ni dia adalah bawah kategori antimicrobial protein. Okay. But the interferon is produced by the host cell kan? It's produced by the host cell against by the infected body cell. Okay. So interferon is produced by infected body cell. So virus infected the host cell. So it triggers a gene to be activated that produces mRNA, and from the mRNA it produces the protein which is interferon. So next interferon will migrate to the healthy cell in uninfected cell. So when interferon enters into the healthy cell, the uninfected cell, it triggers a gene to be activated. So this gene akan menghasilkan the next protein which is antiviral protein. This antiviral protein will prevent virus from getting or entering into the infected body cell. So it limits cell to cell spread of the virus. Kan? So itu di bawah dia fall under antimicrobial protein. Another type of antimicrobial protein adalah apa dia? Complement system. Okay, ataupun complement proteins. So itu akan tengok macam mana awal is complement protein ataupun complement system dia itu ada dalam awak punya lecture note kan. So complement protein adalah a set of protein when it finds or come across a virus or pathogen, dia akan insert into the plasma membrane of the virus or the pathogen, forming pores. Kan? So once pores is produced on the plasma membrane of the pathogen, kan? So maksudnya ada lubang-lubang dekat plasma membrane of the pathogen tu. So then water or ion can enter into the virus or pathogen and causes the virus to swell and then burst, lyse, pecah mati. So that is complement system. So ingat antimicrobial protein dalam second line terbahagi kepada interferon dan juga complement system. In this case, protein U ni adalah interferon lah. Okay, so give one function of protein U. Tadi Fatih, so Najib. So tadi saya ada bagi satu jawapan. So what are the function of the interferon?
To activate the gene for antiviral protein production.
Boleh.
In other cell.
Boleh lah. Okay. So to activate the gene in uninfected cell to produce antiviral protein. What is the function of antiviral protein? The next point, which is to prevent or limit cell to cell spread of virus. Point yang lain awak boleh letak. Ataupun inhibit viral reproduction. So kalau macam soalan ni dia bagi dua markah ke ataupun give two function of U, boleh tambah lah kat situ kan. Inhibit viral reproduction, cause uninfected cell to produce antiviral protein. Yang ketiga, limit cell to cell spread of the virus, and then interfering with the virus ataupun help activate macrophage to enhance phagocytosis. So ada a few function of antiviral protein. Interferon actually. Okay. So thank you, Najib. Next question. C.
Complete the table below to show the differences between B and T cells. So for B cells, the origin of the cell is bone marrow.
Okay.
And for T cell, the site of cell maturation is thymus gland.
Betul. Thymus gland. Okay, thymus. So thank you, Zahratul. Next, Adlin. D.
Figure 7 shows an interaction between the cells involved in the activation of B cells in humoral immune response. Number 1, identify cell Z. Cell Z is T helper cells.
T helper cell ataupun TH, okay. T helper cell. Betul. Okay, so next, Anissa.
Explain how cell Z is involved in the activation of B cell. The TH cell are activated when their receptor combine with foreign antigen MHC. Antigen complexes on APC. Activated TH cell recognize antigen MHC complex on the B cell, and TH cell secrete cytokines to activate B cell.
So, B cell when it is activated, dia akan undergo proliferation and then produce two clones, which is effector and also memory cell. Boleh tambah kat situ. Okay, activated B cell differentiate into plasma cell and also memory cell. So sama juga macam gambar yang tadi kan. Cuma tadi dia buat untuk cytotoxic T cell. Soalan ni dia guna untuk B cell. Sama je activation. Yang penting mesti ada helper T cell to produce and secrete cytokines. The cytokines produced by the helper T cell is the signaling molecule that will trigger the activation of B cell or the activation of cytotoxic T cell. Okay, and also it also triggers the helper T cell to also be activated and produces clones, which is effector and also memory. So bila sel-sel lymphocyte ni activated, dia akan menghasilkan effector and also memory. Okay. So tadi dah betul maka jawapan awak tadi. Okay, E. Question E. Arina.
Explain what is meant by the term infectious disease. Infectious disease caused by pathogenic microorganisms such as bacteria, virus, parasite and fungi.
Okay, betul. Dapat satu markah. So soalan definition ni selalu satu markah je. Okay. So next, Hanis. Number 2.
Give the scientific name for the vector that causes dengue. Aedes aegypti, female mosquitoes.
Okay, Aedes aegypti and also Aedes albopictus. Ada dua So make sure bila dia suruh bagi scientific name, you write, kalau awak handwritten mesti underline. Tapi kalau awak type, mesti italic. Kalau dapat nama dia betul, ejaan dia pun, tapi awak tak ikut procedure writing scientific names, your answer might be wrong. Ingat tu sebab kita dah belajar semester lepas how to write scientific names. So make sure you write the name properly. Walaupun awak dah susah payah ingat ejaan nama mosquito ni, tapi bila awak tulis dia not according to the scientific name format, dia akan menyebabkan jawapan awak boleh jadi salah. Okay. So number 3, Nadia.
TB is another infectious disease in Malaysia. Suggest 2 preventive measures that should be taken to avoid TB. So first, get TB vaccine. Second, improving the ventilation in indoor spaces.
Okay, betul. TB vaccination nak kita panggil dia apa?
BCG.
BCG. Given when?
Babies.
Baby lagi kan. So immunization lah. So immunization by taking BCG masa baby lagi, ataupun improving ventilation indoors. So make sure kalau tak buka tingkap ke, make sure ventilation dalam sesuatu bangunan tu ataupun bilik is ventilated well lah. And then lagi satu boleh letak point, ada a few kan, which is practice hygienic lifestyle, kan, tak batuk depan muka orang atau basuh tangan and all that. Okay, infected people, cough etiquette and respiratory hygiene, yang macam kita amalkan during COVID ni kan. So ada cough etiquette dia, okay. And then using germicidal ultraviolet lamps. So itu pun ada dalam tawak, okay, which is to kill airborne bacteria in buildings, okay. So ada a few yang awak boleh list down, dia nak 2 sahaja, okay. So I guess because it is 4:49 already, so I guess we can end our